Usually when we talk about a medical diagnosis, there's this expectation of precision, right? It feels almost like engineering.
Oh, absolutely.
Yeah.
We want things to be, clear cuts.
Yeah, like you break your arm, the x-ray shows that jagged white line of the fracture, and the doctor just points at the film and says, there it is. That's the problem.
Right. It's a very comforting binary. We really like our biology to be visible, to be neatly categorized as, broken or unbroken.
Exactly.
Yes.
But then you step into the world of neurodevelopment or psychiatric conditions, and suddenly that x-ray machine is just completely useless.
Yeah. Totally obsolete in that context.
I mean, if you go to a psychiatrist for profound autism or schizophrenia or, say, severe depression, there is no blood test.
No.
There is no scan that shows a jagged white line. We are looking at a diagnostic landscape that is just absolute muddy waters. They're diagnosing you based on outward behavior, not the underlying microscopic biology.
Which is incredibly frustrating for both the clinician and the patient, honestly.
Right. So today, we are wading directly into those waters with you, our listener, to clear them up. Because we are taking a journey that starts at the absolute bleeding edge of laboratory neuroscience.
And we mean the literal bleeding edge.
Yeah, I'm talking about literally looking at human brain circuits growing in glass dishes. And we are going to trace that revolutionary science all the way back to the practical, daily, second-by-second rhythms of your own body.
It's a massive leap, but the connection is, it's profound.
It really is. Our mission today is to explore how scientists are actually beginning to cure some of the most complex, devastating conditions known to humanity. But the crazy part is, the underlying mechanisms of these laboratory cures hold the exact same secrets to optimizing your own mental health, your focus, and your brain power right now.
The bridge between that microscopic world of clinical pathology and the everyday reality of human performance is, well, it's absolutely stunning once the mechanisms are laid bare.
Can't wait to unpack it.
We are fundamentally talking about the architecture of the human mind and how it literally wires itself.
So to map this out for you today, we are pulling from a really phenomenal stack of sources. We're looking deeply at the pioneering research from Dr. Sergiu Paisica's Stanford Brain Organogenesis program.
Groundbreaking stuff.
Yeah, totally. And then we're layering in insights from leading neuroscientists on this miraculous little molecule called brain-derived neurotrophic factor, or BDNF.
The brain's master chemical.
Exactly. And then we are connecting all of that to robust clinical data on the profound immediate impact of structured breathwork on the nervous system, drawing heavily from the work of researchers like Jack Feldman and journalists like James Nestor.
Because the incredible reality here is that scientists in Dr. Paika's lab are taking skin cells, turning them into stem cells, and growing customized functioning human brain circuits in a dish.
Which is wild.
Right, to solve these psychiatric mysteries. But the ultimate hack for accessing and upgrading your own brain circuitry might just be the exact way you are inhaling and exhaling right at this very moment.
That tension is what makes this so compelling to me. It's the highest of high-tech lab science, intersecting with the most ancient, built-in physiological tools we possess.
We carry the tools with us everywhere.
We do. So let's start in the lab. Because we have to kick things off by asking a really fundamental question about how medicine even works.
Right.
If you are a researcher trying to cure a uniquely human psychiatric disorder, say profound autism or schizophrenia, how on earth do you study that in a mouse?
Well, you can't. And that is the historic bottleneck of all psychiatric research. For decades, it has been a profound limitation based on, accessibility and complexity.
Right.
You simply cannot biopsy a live human brain to see what's going wrong in a psychiatric disorder. I mean, if a patient has a liver condition, a hepatologist takes a needle, extracts a tiny core of the liver, puts it under a microscope, and has an immediate biological answer.
But the brain is a different story.
Exactly. The human brain is locked inside a thick vault of bone, and any tissue you extract causes permanent, irreversible cognitive or physical damage to the patient. So the scientific community relied on animal models, predominantly mice.
Which makes sense for basic biology, sure. I mean, a mouse has a heart, lungs, a liver.
Yeah, the basic plumbing is similar.
But a mouse is not a human when it comes to the mind.
Not even close. While mice have been invaluable for understanding basic cellular functions or very simple behavioral responses, they fail spectacularly when modeling human psychiatric conditions.
Because they just don't have the hardware.
Right. A mouse does not possess the incredibly complex, highly expanded cortical circuitry that gives rise to human language, existential dread, complex social behavior, or the sophisticated cognitive deficits we see in a disease like schizophrenia.
They just don't have the wrinkles.
Exactly. The human cerebral cortex is deeply folded. All those wrinkles and grooves called sulci and gyri, that's what allows us to pack billions of neurons into our skulls. A mouse brain is smooth.
Yeah.
It lacks the architectural real estate. I mean, you can use genetic engineering to mutate a gene in a mouse that is linked to autism in humans. That mouse might show some altered behavior.
Like what?
Maybe it doesn't sniff other mice as frequently, or it hides more. But is that mouse experiencing the equivalent of human profound autism? The underlying neural architecture just isn't there to support that comparison.
Which is what makes Dr. Sergi Pica's groundbreaking work so unbelievable. To get around this mouse problem, his team at Stanford essentially created a sandbox environment for human brain development.
A living sandbox.
Yeah. When I read about the mechanics of this in the sources, it honestly felt like science fiction. Because they aren't taking brain tissue from patients. They are taking regular human skin cells.
Fibroblasts.
Right, just a tiny superficial skin biopsy.
And this entirely relies on a historic Nobel Prize-winning discovery regarding what are called Yamanaka factors. Scientists learned that you can take a mature, fully differentiated adult cell like a skin cell and expose it to a specific cocktail of 4 proteins.
So they just wash the skin cell in these proteins?
Essentially, yes. This chemical bath wipes the cell's memory. It chemically induces the fibroblast to revert back into its embryonic state. It becomes what is called an induced pluripotent stem cell.
Pluripotent, meaning it has the potential to become absolutely any tissue in the entire human body.
Exactly. It is a biological blank slate.
Okay, so you have this blank slate in a dish.
Yeah.
A microscopic lump of pure potential. How do they get it to become a brain? I mean, they don't just leave it there and hope it figures it out, right?
No. They have to act as the biochemical directors of the cell's fate. They provide a very precise sequence of growth factors and nutrients that mimic the exact chemical environment of a developing human fetus.
Wow, they're faking fetal development in the dish.
They are. They coax these blank slates down the developmental pathway to become neural progenitor cells. But the critical innovation from Pica's lab wasn't just making the cells, it was how they grew them.
Right, because historically, didn't scientists just grow cells in a flat, two-dimensional layer on the bottom of a plastic Petri dish?
Yes, like a cellular pizza.
A cellular pizza.
A flat, chaotic web of cells. The problem is, a flat layer of neurons doesn't represent the spatial geography of a real three-dimensional human brain.
Because the brain isn't flat.
Exactly. So Pica's team utilized special plates that prevent the cells from sticking to the bottom. They kept the cells suspended in a nutrient-rich liquid.
Oh, so they're just floating there.
Right. And because the cells couldn't attach to the plastic, they clung to each other. They formed a floating three-dimensional sphere. And when allowed to grow in this 3D suspension, something magical happens. The cells self-organize.
They know what to do on their own.
Yes. They follow their own internal genetic instructions to form concentric layers, mimicking the actual distinct layers of the human cerebral cortex. They created what we call a cortical organoid.
A miniaturized, living 3D model of human brain tissue. That is insane.
It really is.
But they didn't stop there. They took these organoids and created what they call assembloids.
Well, because an organoid on its own is isolated. It's like a neighborhood with no roads connecting it to the rest of the country.
Right.
An assembloid is created when you take 2 different organoids. For instance, one that was guided to become the cerebral cortex, and another that was guided to become a deeper brain structure, like the striatum, or even the spinal cord.
Okay, and then what?
You physically place these 2 distinct 3D spheres next to each other in the culture dish, and over the course of a few days, they physically fuse together.
And they don't just stick together like 2 lumps of clay.
Right.
The sources said the cells actually start communicating.
They wire together. The neurons from the cortical organoid will sprout long projections called axons.
Reaching out.
Exactly. Guided by chemical attractants released by the other organoid, these axons physically reach across the microscopic gap, find their specific target neurons in the striatum organoid, and form functional firing synapses.
That's unbelievable.
They start sending electrical signals back and forth. You are quite literally watching a customized human neural circuit build and wire itself in real time, completely outside of a human body.
Okay, so let's look at what this technology actually reveals about a condition like autism. Because for a very long time, I feel like the public perception, and maybe even the baseline medical assumption, was that in severe neurological conditions, there was just a structural deficit.
A missing piece.
Yeah, like a chunk of the brain simply failed to form, or millions of cells just died off and left a void.
And those assumptions are exactly what these assembloids are systematically disproving, particularly regarding neurodevelopmental disorders.
Oh, really?
Yes. What Panka's lab is revealing at the microscopic level is that autism is almost certainly not a case of missing brain cells.
So the cells are there?
Yes. If you take skin cells from a patient with profound autism, revert them to stem cells and grow an organoid, you see that the neurons are generally all there. The tissue mass is there, the cells are alive, and they're generating electrical voltage.
Wait, if the cells are all present and accounted for, where is the disease living?
It lives in the connectivity and the timing. The hardware components are there, but the way they wire up, the speed at which the neurons migrate to their final structural destinations, and the synchrony with which they communicate are subtly but profoundly altered.
So it's a software and wiring issue, not missing hardware.
Think of a healthy neural circuit like a beautifully orchestrated symphony. Every section comes in at the exact right microsecond. In some of these models of autism or schizophrenia, the firing is chaotic. The neurons might migrate too slowly during development, missing their critical window to connect with a specific partner, or the connections they do form are wildly imbalanced, too strong in some regions and far too weak in others.
That structural reality explains so much of what we see clinically. I mean, if it's a systemic wiring and timing issue, it makes perfect sense why autism is such a broad spectrum. And crucially, why it so frequently presents with other devastating co-occurring conditions. The sources note that profound autism is often accompanied by significant intellectual disability, or intractable epilepsy, which is, severe uncontrollable seizures.
Yes, that overlap is very common.
So it's not three different diseases. It's the exact same underlying circuitry imbalance manifesting in different ways across the brain's vast network.
Precisely. When you look at local circuits in the cortex, they rely on a delicate balance between excitatory neurons, which shout fire, and inhibitory neurons, which say quiet down.
The gas pedal and the brake.
Exactly. If the genetic instructions cause that local circuit to miswire, resulting in too much excitatory activity and a lack of inhibitory control, that doesn't just alter how the brain processes social cues or sensory input.
Right.
It physically manifests as an electrical storm, a seizure. Being able to study the exact living circuitry of a specific patient who suffers from these seizures, all safely contained in a dish, is a total paradigm shift for medicine.
And we have to highlight a massive historic triumph from the sources regarding a very specific disease called Timothy syndrome.
Oh, this is an incredible case study.
It really is.
Yeah.
So Timothy syndrome is this rare, devastating genetic condition. Children born with it experience profound autism and life-threatening epilepsy. Panka's lab took skin cells from these patients, grew them into assembloids, and looked at the microscopic wiring.
Because they had the living circuit right in front of them.
Yes, and because they could actually watch it run, they found the exact molecular defect, and unbelievably, they found a way to drug it.
The mechanics of this breakthrough are astonishing. Using these patient-specific stem cell models, they zoomed in on the cellular membranes.
Okay.
Neurons communicate by firing electrical action potentials, and they do this by opening and closing tiny protein gates called ion channels. These channels let charged particles like calcium flow in and out of the cell.
Like tiny doors?
Yes. In Timothy syndrome, the researchers discovered that a genetic mutation was causing a very specific calcium channel to stay open just a fraction of a millisecond too long.
Just a millisecond, like a gate on a dam getting stuck, letting too much water rush through.
Precisely like that. And that microscopic delay allowed too much calcium to flood into the neuron. Now, calcium isn't just a mineral for your bones. Inside a neuron, it is a potent signaling molecule.
So the flood causes chaos.
Massive chaos. That massive calcium overload altered the neuron's voltage, caused the cells to pull back their dendritic branches, and triggered the faulty wiring and electrical storms that led to the seizures and the autism phenotype.
Wow.
But here's the brilliant part. Because the scientists had thousands of these living diseased assembloids in the lab, they could rapidly test thousands of different chemical compounds on them.
Trial and error at scale.
Exactly. They systematically searched for a molecule that would bind to that specific calcium channel and force it to close on time. And they found it.
They actually found it.
They identified small molecules that corrected the defect, reversed the abnormal wiring in the dish, and they are now moving toward clinical trials.
That gives me chills. This will be the very first therapeutic for a psychiatric disease developed exclusively using human stem cell models. They bypass the mice entirely.
It's a monumental shift in how we do pharmacology.
But, as incredible as this is, I'm listening to this and a massive question pops up.
What's that?
If Dr. Pakai can literally grow my customized neural circuit in a dish, figure out exactly which calcium channel is stuck, and test thousands of drugs on it, why aren't we curing schizophrenia tomorrow? What is the bottleneck in this biological sandbox?
It's the essential question, and the answer lies in the staggering complexity of the brain's emergent properties. The brain is not just a collection of isolated local circuits. It is a vast, long-range, interconnected geography.
Meaning it's bigger than just one neighborhood?
Much bigger. In complex psychiatric conditions like schizophrenia, the profound cognitive deficits often arise from faults that happen across massive physical distances in the brain. A neuron in the prefrontal cortex, the area responsible for logical thought and executive function, has to communicate perfectly with a neuron deep in the amygdala or the hippocampus.
Right.
That single neuron has to send a physical projection, an axon, that spans several inches across the brain tissue.
Which, if you scale it down to the size of a single microscopic cell, is an unbelievable distance. It's like a fiber optic cable stretching from New York to London.
The New York to London analogy is perfect. Assembloids are brilliant for showing us local connectivity, how one neighborhood in New York talks to the neighborhood right next door.
Right.
But scientists are still struggling to build models that replicate those massive, long-range, brain-wide synchronized connections. You can't easily model a transatlantic cable in a tiny glass dish.
That makes a lot of sense.
And furthermore, an organoid floating in a nutrient bath lacks environmental inputs.
It's living in a bubble.
Literally. It doesn't experience childhood trauma. It doesn't experience the complex cocktail of puberty hormones. It doesn't interact with the human immune system. And it doesn't experience sensory overload.
Oh, wow. Yeah.
Those external lived experiences heavily influence how a psychiatric disease actually manifests. The dish perfectly reveals the biological hardware defect, but it cannot show us the software bugs that develop through a lifetime of a human being interacting with the world.
Which is such an important limitation for us to understand. And sadly, that exact gap between what we want stem cells to do right now and what they are actually capable of doing brings us to a much darker reality highlighted in the sources.
Yes, the exploitation of hope.
Exactly. Because when the general public hears about these miraculous, Nobel-winning stem cell breakthroughs, it creates a massive, desperate hope. And sometimes that hope is dangerously exploited.
The sources outline a very sobering reality regarding the rise of unregulated stem cell tourism.
And we need to be incredibly objective here. We are strictly reporting the biological facts as presented in the source material. We aren't taking sides. But the sources highlight that there are desperate parents taking children with profound autism or adults with neurodegenerative diseases to unregulated, unproven clinics, often overseas.
Searching for a miracle.
Right. They are paying tens of thousands of dollars out of pocket to have mysterious, undefined stem cells injected directly into their spinal fluid or their bloodstream, operating under the assumption that these magical cells will just swim up to the brain and cure the autism.
And scientifically, neurologically speaking, these procedures lack almost any biological justification.
It's just not how it works.
We have to clearly explain the underlying mechanics of why injecting stem cells into a living patient simply does not work for complex neurodevelopmental disorders.
Let's unpack that deeply, because intuitively, to a layperson, it makes sense, right? If the brain's wiring is broken, why can't we just inject fresh, healthy, pluripotent stem cells to go in there, act as a microscopic repair crew, and lay down new wires?
It all comes down to a biological concept called the permissiveness of the environment.
Permissiveness, okay.
We have to compare the fetal brain to the adult brain. During early fetal development, the brain is an incredibly permissive, dynamic construction zone.
It wants to grow.
Exactly. The environment is flooded with highly specific chemical gradients, trails of molecular breadcrumbs that guide migrating cells.
Okay, breadcrumbs.
And there are specialized structures called radial glia that act like physical ropes, extending from the deep center of the developing brain all the way to the outer cortex. Baby neurons physically wrap around these ropes and crawl up them to reach their final destination.
Wow, literally climbing ropes.
The environment is actively, eagerly facilitating growth and long-range connection.
But once we are born, and certainly by the time we are adults, that construction zone shuts down. The ropes are gone.
The radial glia disappear entirely. The chemical breadcrumb trails fade away. The adult brain, and even a young child's brain, once that massive initial wiring phase is complete, becomes stubbornly, defensively rigid when it comes to long-range structural changes.
It sounds like trying to pour fresh, wet cement into a skyscraper that is already fully built, completely occupied with office workers, and hoping that the wet cement somehow magically forms a brand new, perfectly placed, load-bearing pillar.
That is exactly what happens. You cannot just inject a naive stem cell into the spinal fluid and expect it to magically know what to do in an adult environment.
Because there's no blueprint anymore.
Exactly. It has no radial glia to climb, it won't know where to migrate, it won't know how to grow a projection down through millions of densely packed existing connections, somehow bypass the immune system, and seamlessly join an established, complex cortical circuit.
The office workers are going to reject the wet cement.
Not only does the wet cement not form a pillar, but it disrupts the entire building. The brain's immune system, the microglia, acting as security guards, will likely recognize these injected cells as foreign invaders and attack them, causing massive inflammation.
Oh, wow.
Or worse. Because pluripotent stem cells are programmed to endlessly divide and grow, and the adult brain lacks the stop signals of a fetal brain, those injected cells can just keep growing until they form devastating tumors called teratomas.
That is absolutely terrifying. The intention is a cure, but the intervention is a biological sledgehammer swinging in the dark.
It's tragic, honestly.
And this rigidity, this lack of permissiveness in the adult brain, explains so much more than just the failure of stem cell tourism.
Yes, it's a fundamental principle of neurology. It is the core reason why traumatic brain injuries, severe strokes, and spinal cord injuries are so permanent and devastating.
Because it just won't regrow.
Right. As the researchers note, once human cortical neurons are lost or severed, they are largely gone forever. Unlike your skin or your liver, which can regenerate after an injury, we do not have massive hidden reservoirs of stem cells in our brains waiting to replace lost cortical tissue.
So there's no backup supply.
We do generate a tiny microscopic trickle of new neurons in very specific small regions like the hippocampus, but you are absolutely not regenerating your prefrontal cortex after a major stroke.
So this leaves us at a fascinating biological crossroads for you and me. If we cannot safely add new cellular hardware to the brain, and we cannot build brand new long-range neurological highways in adult using stem cell injections, how do we actually fix or upgrade the circuitry we already have?
Right. If you can't build a new building.
Exactly. If we are stuck with the skyscraper we've built, how do we remodel the offices inside to make them run better?
This is where the science pivots away from external cellular interventions and focuses entirely on the brain's internal built-in maintenance systems.
The software updates.
Yes. If we can't add new hardware, we have to maximize the software and the existing connections. We have to dive into the architecture of neuroplasticity, and specifically, we have to talk about its master chemical controller.
Okay, so if the brain's intricate web of circuits is a vast garden, we are about to introduce the industrial-strength, highly targeted fertilizer. Let's unpack a molecule called brain-derived neurotrophic factor, universally known as BDNF.
Neuroplasticity, the ability of the brain to literally change its physical wiring and strengthen its connections in response to learning, experience, or injury, isn't just some abstract psychological concept.
It's an actual physical process.
It is a tangible, mechanical, chemical reality driven by specific proteins. BDNF is arguably the most critical player in this process. It is a neuropeptide, part of a family of proteins known as neurotrophins.
So where does this master chemical actually do its work? Where is the fertilizer applied?
While it is found throughout the central nervous system, it is most heavily concentrated in two vital areas. First, the hippocampus, which is the brain's primary hub for learning, memory consolidation, and spatial navigation.
Okay.
And second, the cerebral cortex, which handles our higher-level executive functions, decision-making, and complex thought.
The really important human stuff.
Exactly. On a microscopic level, BDNF is responsible for the actual physical growth of new dendrites, the branch-like structures that neurons use to receive signals.
Like growing new branches on a tree.
Yes. It ensures the survival of existing neurons, and critically, it drives a process called long-term potentiation, which is the physical strengthening of the synaptic connections between two neurons that fire together frequently.
So if you are trying to learn a new language, or memorize a complex physical skill like playing the piano, or even trying to recover cognitive function after a trauma, you biologically require BDNF to cement those new neural pathways into place.
You cannot learn without it.
But the sources dive into some incredibly counterintuitive nuance here. It is not as simple as more BDNF equals a universally better brain.
Mm-hmm.
The molecule has a dual nature.
A dark side and a light side, almost.
Right. There is pro-BDNF and there is mature BDNF.
This duality is crucial for understanding the immense delicacy of brain chemistry. BDNF does not pop out of the cellular machinery in its final helpful form. It is initially synthesized as a larger precursor molecule called pro-BDNF.
Okay.
Now, when the brain enzymes cleave it and turn it into mature BDNF, that mature molecule binds to a very specific receptor on the surface of neurons called the TRKB receptor, pronounced TRKB. When mature BDNF docks into the TRKB receptor, it flicks a biochemical switch inside the cell that screams, grow, thrive, build stronger connections.
That's the miracle grow effect.
Exactly. But evidence demonstrates that the larger precursor, pro-BDNF, can also be secreted by cells. And pro-BDNF doesn't fit into the TRKB receptor.
He goes somewhere else.
It binds to a completely different receptor called the P75 receptor. And when pro-BDNF binds to P75, it initiates the exact opposite cascade.
The opposite.
It weakens synaptic connections. It tells the dendritic branches to shrink and pull back. It can even trigger a process called apoptosis, which is the highly regulated programmed death of the cell itself.
Wait, let me make sure I'm getting this. Why on earth would the human brain evolve to actively produce a chemical that explicitly tells its own cells to weaken or commit suicide? That sounds like a catastrophic design flaw.
It does sound counterintuitive, but on the contrary, it is a brilliant and necessary design.
How so?
Let's go back to the garden analogy. If you only ever apply fertilizer and you never use pruning shears, what happens?
Oh, I see. You get a jungle.
You get an overgrown, tangled, hyperdense, choking mess of vines where no sunlight gets through and the system fails. A hyperconnected brain is a noisy, chaotic, inefficient brain.
You remember every single useless detail of your day.
And you wouldn't be able to process any of it. The brain absolutely needs to be able to strengthen the important, frequently used connections, like the route you drive to work every day. But it equally relies on pruning away useless, inefficient, or out-of-date connections to keep the overall network fast, streamlined, and energetically efficient.
So pro-BDNF and the P75 receptor act as the biological pruning shears to balance out the miracle grow.
Exactly. It's a constant dynamic balance.
That makes total sense. You have to weed the garden. But here's my fundamental question. If mature BDNF is this undeniable chemical powerhouse that fights clinical depression, stabilizes mood disorders, and heavily enhances memory, and the sources explicitly point out that standard antidepressant medications like SSRIs ultimately achieve their long-term effects by increasing BDNF signaling, why is this not a supplement?
Ah, the magic pill question.
Right. Why hasn't the pharmaceutical industry just manufactured BDNF in a pill that I can take with my morning coffee to guarantee a super brain?
It is the ultimate neuroenhancement dream, but it fails against several massive biological brick walls.
What's the first wall?
The first wall is the blood-brain barrier, or the BBB. The blood vessels that feed your brain are not like the blood vessels in your arm.
How are they different?
In your arm, the cells lining the blood vessels have tiny gaps between them, allowing nutrients and chemicals to easily slip out into the surrounding tissue. In the brain, those endothelial cells are fused together by tight junctions, and they are further wrapped in the protective arms of cells called astrocytes.
Like a fortress.
It creates an incredibly selective, highly fortified biological wall. BDNF is a relatively large, complex protein. If you swallow it in a pill, your stomach acid will obliterate it instantly.
Okay, so a pill is out.
But even if you injected it into your bloodstream, that massive protein cannot squeeze through the tight junctions of the blood-brain barrier to actually reach your neurons.
Okay, so let's play mad scientist. What if we drill a hole and inject the BDNF directly into the cerebrospinal fluid, bypassing the barrier entirely?
Even if we overcome the delivery problem, we run into the second, far more dangerous problem: timing and location.
Timing and location.
Artificially flooding the entire global architecture of the brain with mature BDNF is a terrible idea because widespread, untargeted synaptic growth is dangerous. You do not want to strengthen every connection in your brain.
Because it's a jungle again?
Yes. BDNF is agnostic. It has no moral compass. It simply strengthens whatever neural circuits happen to be highly active at the exact moment it is released.
Oh, wow.
So if you artificially flood your brain with BDNF and you happen to be experiencing a severe traumatic event, or you are deeply engaged in an addictive behavior, you will permanently, heavily hardwire that trauma or that addiction deep into your circuitry.
That is terrifying.
Therefore, the exact timing of the release and the highly specific microscopic location of the release are everything.
So taking a global BDNF pill is like indiscriminately dropping fertilizer out of an airplane over the entire state of California, hoping it makes your specific tomato plant grow, but accidentally creating a massive superforest of invasive, destructive weeds everywhere else.
That is precisely the danger. You want the fertilizer applied exclusively to the microscopic roots of the specific neural pathways you are actively trying to cultivate.
Which brings us to the empowering part of this science for everyone listening. If we can't safely pill it, how do we trigger it naturally? Because the sources are emphatically clear that we have immense conscious control over our own internal BDNF production.
We absolutely do. And the most robust, scientifically validated, undeniable way to dramatically increase BDNF production in the human brain is aerobic exercise.
Just getting your heart rate up.
The clinical data in the sources notes that engaging in a consistent hour-a-day aerobic exercise regimen can literally quadruple the baseline release of BDNF from your brain within just 3 months.
Quadruple.
Yeah.
A 400% increase just from sweating.
Yes.
And another major trigger highlighted in the research is intermittent fasting, or placing the body in a temporary caloric deficit. But we have to explain the mechanism here. Why does the brain look at physical exertion or a grumbling stomach and decide that it's the perfect time to upgrade its cognitive hardware?
To understand the mechanism, you have to view it through the lens of our evolutionary history. The biology makes elegant sense.
How so?
Under what exact conditions were our ancient ancestors experiencing severe, sustained physical exertion and a negative calorie balance at the exact same time?
Well, when they were starving and having to track down prey over miles of terrain, or when they were sprinting to avoid becoming prey themselves.
Exactly. In times of profound physical stress or caloric scarcity, survival was immediately on the line. That was absolutely not the time for the brain to be sluggish, depressed, or foggy.
Right. You can't have brain fog when a tiger is chasing you.
Our ancestors needed to be cognitively razor-sharp, highly focused, and capable of rapidly learning new details about their environment, like a new hunting route or the location of a predator, just to survive the day.
So the stress was a signal.
Therefore, metabolic stressors act as direct chemical messengers. When you exercise intensely, your muscles produce lactate. When you fast, your liver produces ketones. These molecules travel through the blood, cross the blood-brain barrier, and physically signal the hippocampus to start churning out BDNF. The body is essentially screaming to the brain, resources are scarce, the environment is hostile, I need you firing on all cylinders, adapting instantly and remembering everything that happens right now so we don't die.
That is profound. Our underlying biology literally views physical effort and hunger as a mandatory signal to trigger neuroplasticity.
It's an ancient survival loop.
But there is a massive catch to all of this. Most of us listening to this right now aren't running down antelope on the savannah. We sit hunched at desks, staring at glowing rectangles.
Very true.
And while diet and exercise are phenomenal tools for boosting BDNF, there is a much more immediate, deeply foundational biological rhythm that constantly dictates our brain state. And most of us completely ignore it.
We take it totally for granted.
You can exercise all you want, but if your autonomic nervous system is chemically trapped in a state of survival and panic, the gates to positive neuroplasticity remain firmly locked.
This is a critical pivot. We are talking about the breath-brain connection.
The most overlooked trigger for mental health. We obsess over swallowing the perfect omega-3 supplements. We track our 10,000 steps. We meticulously measure our macros. But we breathe between 20,000 and 25,000 times a single day.
Every single day.
And according to the sources, specifically leaning into the deep physiological research highlighted by Dr. Jack Feldman and the journalistic synthesis by James Nestor, modern humans are breathing completely, fundamentally wrong. And it's actively destroying our neurological health.
It is a hidden systemic epidemic. The sources describe modern dysfunctional breathing as fast, shallow, apical meaning into the upper chest rather than the diaphragm, and predominantly through the open mouth.
Mouth breathing.
The vast majority of us are chronically overbreathing, essentially existing in a low-grade state of hyperventilation all day long while we answer emails, sit in traffic, or scroll on our phones.
Okay, we need to mechanically unpack why overbreathing is such a disaster. Why does breathing a little too fast completely derail the brain?
It all comes back to the massive communication highway running between the body and the brain, which is primarily mediated by the vagus nerve.
Right, the vagus nerve.
We often think the brain is the dictator telling the body what to do. But realistically, 80 to 90% of the nerve fibers in the vagus nerve are afferent, meaning they travel from the organs up to the brain.
Wait, really? 90%?
Yes. The brain is locked inside a dark, silent bone box. It has no idea what is actually happening in the outside world. It completely relies on sensors in the body to tell it whether we are safe or in mortal danger.
So when you are hunched over your laptop, mouth breathing fast and shallow into your collarbones,
you are manually sending a mechanical alarm signal straight up the vagus nerve to the brainstem. Fast, shallow breathing perfectly mimics the physiological state of a panic attack or a full-blown fight-or-flight response.
Oh, wow. So you're tricking your brain into panicking.
Your brain receives those rapid, rhythmic signals from the diaphragm and the lungs and logically assumes we must be running from a tiger right now. It immediately shifts your autonomic nervous system into a sympathetic, high-alert state.
Distress state.
It unleashes a cascade of adrenaline and cortisol into your bloodstream. If you breathe this way, chronically, all day, every day, it leads directly to systemic inflammation, chronic anxiety, and profound brain fog. Your brain is exhausted because, chemically speaking, it thinks it has been fighting for its life for 10 hours straight.
And here is where we hit one of the most counterintuitive, mind-bending facts in our entire stack of research, the CO2 paradox.
Oh, this is fascinating.
Because my entire life, I have been taught a very simple elementary school equation. Oxygen is the good, life-giving energy molecule. Carbon dioxide is the bad, toxic waste product that we must aggressively expel from our bodies.
We all learn that.
But the clinical sources, particularly those studying the Buteyko breathing method, completely shatter that assumption.
They systematically dismantle it. The vilification of carbon dioxide is one of the greatest misunderstandings of basic respiratory physiology.
Why?
Yes, CO2 is a byproduct of cellular metabolism, but it is also a master, non-negotiable regulator of your entire vascular system. When you hyperventilate, when you breathe through your mouth and overbreathe, you blow off far too much carbon dioxide.
You exhale too much of it.
The CO2 levels in your blood drop below the healthy baseline.
But why is low CO2 a bad thing? If I'm breathing fast, I'm pulling in tons of oxygen, right? Shouldn't my brain be flooded with life-giving energy?
This is where we have to explain a mechanism called the Bohr effect. Oxygen is carried through your bloodstream by a protein called hemoglobin, which sits inside your red blood cell.
The delivery trucks.
Exactly. But hemoglobin acts like a very stubborn, very strict delivery driver. It will pick up the oxygen in the lungs, but it refuses to just drop the oxygen off to your tissues arbitrarily.
It holds on to it.
It requires a specific biochemical signal to release its cargo. That signal is the presence of carbon dioxide.
Yeah.
As your cells work, they produce CO2. This CO2 slightly lowers the pH of the blood in that local area, making it slightly more acidic. When hemoglobin encounters that slightly acidic CO2-rich environment, it physically changes its shape, loosening its grip and finally releasing the oxygen into the cells of your brain and body.
Wow.
Furthermore, carbon dioxide is a potent vasodilator. It actively relaxes the smooth muscle around your blood vessels, forcing them to widen and open up.
So let me use an analogy here to make absolutely sure I'm visualizing this correctly. It's like oxygen is a VIP guest trying to get into the exclusive nightclub of the brain cells.
I love this analogy.
But oxygen isn't on the list, and it can't open the door itself. Carbon dioxide is the massive bouncer standing at the door. If the CO2 bouncer isn't there, the door stays shut, and oxygen just floats right past the club in the bloodstream, completely unable to enter the party.
That is a wonderfully accurate visualization. If you have low CO2 from overbreathing, two utterly disastrous things happen to your brain simultaneously.
Yeah, what's the first?
First, without the vasodilating presence of CO2, your blood vessels physically constrict, dramatically reducing the overall volume of blood flowing up into your neocortex, the thinking rational part of your brain.
So less blood gets up there, period.
Second, whatever blood does make it up there is full of oxygen, but the hemoglobin holds onto that oxygen with a death grip. It refuses to release it because the CO2 bouncer isn't there to open the door.
Oh my God.
You can be taking huge, massive, desperate gasps of air through your mouth. Your blood oxygen saturation can read at 100% on a pulse oximeter, and yet your actual brain cells are literally suffocating.
That's crazy.
This massive lack of cellular oxygen is the exact physiological cause of the brain fog, the dizziness, and the lightheadedness you feel when you panic or hyperventilate.
So to fix this suffocating brain fog, we have to gently, deliberately accumulate CO2. By breathing slowly, lightly, and strictly through the nose, we keep the bouncer at the door, the blood vessels dilate open, the hemoglobin releases its grip, and oxygen actually floods into the brain cells.
Yes.
It is completely backwards from what intuition tells you.
It really is. And the mechanical connection between breathing and the brain goes even deeper anatomically.
Yeah.
We have to introduce a tiny, miraculous structure called the pre-Butzinger complex.
The pacemaker of breath.
Exactly.
Yeah.
Deep inside the brainstem, there is a tiny, highly specialized cluster of a few thousand neurons called the pre-Butzinger complex. This is the central biological oscillator that generates the rhythmic firing of your diaphragm.
It just ticks away.
It is firing right now, totally subconsciously to keep you alive. But what Dr. Jack Feldman's pioneering research revealed is that this complex doesn't just passively drive the lungs. It acts as an active emotional steering wheel for the entire brain.
How does a breathing pacemaker steer emotions? I mean, how does that physical link work?
The pre-Butzinger complex sends direct physical neuronal projections into other critical areas of the brain, most notably a region called the locus coeruleus.
Okay, the locus coeruleus.
The locus coeruleus is the brain's primary source of norepinephrine, which is the neurotransmitter responsible for regulating global arousal, panic, attention, and focus.
Ah, the arousal center.
Every single time you alter the rhythm of your breath, whether you speed it up, slow it down, or hold it completely, the pre-Butzinger complex fires a message directly into the arousal centers of your brain, dictating exactly how it should feel.
Wow.
If you forcefully change the physical rhythm of your breath, you instantly chemically alter the background emotional state of your entire brain.
Which perfectly, beautifully sets up our final exploration. Because we've traveled from Dr. Paika's stem cells wiring up in a dish to the miracle grow mechanisms of BDNF to the microscopic mechanics of the lungs and the blood. Now, how do we synthesize all of this into action?
The practical application.
Exactly. How do you, the person listening to this right now, use your breath to actively trigger plasticity, forcefully calm your nervous system, and optimize your brain?
We have to bring all the threads back to BDNF and neuroplasticity. We established that the brain absolutely needs to feel safe to undergo targeted positive plasticity. You can't rewire effectively while running from a tiger.
Right.
But we also established that the brain responds to mild, controlled, metabolic stress-like exercise or fasting by releasing a massive surge of BDNF. The sheer beauty of controlled breath work is that it allows you to manually manipulate both of these states on command.
The sources highlight the powerful impact of short breath holds and controlled mild hypoxia, meaning a low oxygen state. When you practice specific, controlled breath-holding techniques, you are intentionally allowing your internal CO2 levels to rise significantly and your blood oxygen to gently, temporarily dip.
And what does the brain do when it senses that mild, highly controlled hypoxic state? It initiates an ancient survival response.
Thinks it's working out.
It panics just a tiny microscopic bit, but in a hermetic, beneficial way. To ensure survival through what it perceives as a harsh environment, the brain starts to release a surge of that BDNF fertilizer.
The miracle grow.
It's essentially signaling the environment is getting challenging. I need to urgently build better neural connections, improve cognition, and solidify memories to navigate this successfully. You are artificially recreating the exact same biochemical trigger as an intense, hour-long run simply by sitting still and manipulating the gases in your lungs.
It is an immediate, accessible shortcut to the miracle grow. But what about the other side of the coin? What about when we are already stressed out of our minds? When the panic is real, when the emails are piling up and our heart is racing? What is the manual override for that?
That brings us to the first actionable tool, Protocol 1, the physiological sigh. This is arguably the fastest, most effective real-time stress killer discovered by modern neuroscience, heavily researched and popularized by Dr. Jack Feldman and Dr. Andrew Huberman.
I use this tool constantly. It's a lifesaver. Let's walk the listener through the exact mechanics of how to do it. It is 2 quick inhales through the nose, followed by 1 long, slow, extended exhale through the mouth. But we need to explain the biological why. Why does that specific pattern, the double inhale, work, like a neurological tranquilizer dart?
It is a purely mechanical rescue mechanism for the tiny structures inside your lungs, which immediately impacts your autonomic nervous system. Deep inside your lungs, you have roughly 500 million tiny balloon-like air sacs called alveoli.
500 million.
If you were to flatten out all the alveoli in your lungs, they would cover roughly the surface area of a tennis court. It is a massive area for gas exchange.
Right.
But because these tiny sacs are lined with fluid, they have surface tension. Over time, especially when we are stressed, sitting for hours or breathing shallowly, those tiny wet balloons start to stick together and collapse.
And when they collapse, they can no longer exchange the oxygen and the carbon dioxide, which raises our internal stress levels even further.
Exactly. The body senses the declining efficiency and starts to panic. When you execute the physiological sigh, that first big, deep inhale fills the lungs almost to capacity. But then you take that second, sharp, smaller inhale right at the very top.
The double sniff.
That second inhale provides a rapid mechanical spike in pressure that forcibly pocks open millions of those collapsed alveoli all at once. It rapidly, violently reinflates the entire tennis court of your lungs.
That is an incredible visual, popping the balloons back open.
Then, with all the alveoli forced open and maximizing surface area, you execute that long, slow, extended exhale through the mouth. That extended exhale does 2 critical things. First, it offloads a massive amount of accumulated, stressful carbon dioxide gas all at once.
Getting rid of the excess.
Second, and more importantly, it strongly mechanically stimulates the vagus nerve.
How does the exhale trigger the nerve?
It's pure anatomy. When you inhale, your diaphragm moves down, creating more space in your chest cavity. Because there is more space, your heart physically expands a little bit, blood moves through it faster, and sensors, called baroreceptors, recognize this speed and tell the brain to speed up your heart rate. But when you exhale, the diaphragm moves up, shrinking the space in the chest cavity. The heart is slightly compressed, blood flows a bit slower, and the baroreceptors instantly signal the brain to slow things down.
The brake pedal.
Yes. When you intentionally make your exhale long and slow, the brain sends a massive signal down the vagus nerve. The vagus nerve literally squirts a neurotransmitter called acetylcholine directly onto the sinoatrial node of your heart, aggressively acting as a brake pedal.
Wow.
In real time, within seconds of that extended exhale, you forcefully shift your entire autonomic nervous system out of sympathetic panic and into a state of profound parasympathetic calm.
It is essentially lifting the hood and hitting the manual override switch on your own anxiety. And the sources note that humans actually do this entirely naturally.
We do.
We involuntarily execute a physiological sigh about every 5 minutes just to keep our alveoli from collapsing.
Yeah.
See dogs do it right before they settle down to go to sleep.
Yeah.
We are just taking conscious, weaponized control of that built-in reflex.
Exactly. We're using it on demand. And once you have used the physiological sigh to kill the acute real-time stress, you can use structured rhythmic breathwork to set the biological foundation for long-term neuroplasticity.
Which brings us to Protocol 2.
Protocol 2, box breathing and the 6-2-8 rhythm.
Box breathing is famously utilized by Navy SEALs to maintain focus and calm in terrifying combat situations. It's inhaling for a count of 4, holding for 4, exhaling for 4, and holding empty for 4, or the other highly effective variation mentioned, the 6-2-8 rhythm. Inhale for 6 seconds, hold for 2, exhale for a long, slow 8 seconds.
The precise seconds matter much less than the underlying mathematical ratio.
Okay, what's the key principle?
The key non-negotiable principle here is the extended exhale. Whenever your exhale is significantly longer than your inhale, you are relentlessly driving that vagal tone. You are continuously squirting that acetylcholine onto the heart, lowering your heart rate, and sending a continuous, undeniable biological signal of profound safety up to the brainstem.
And we have to reiterate why that safety signal is so utterly crucial for BDNF and neuroplasticity. Why can't we just successfully rewire our brains, learn a new language, or process trauma while we are stressed out and hyperventilating?
Because you fundamentally cannot positively rewire a brain that feels it is under mortal attack. When you are locked in a sympathetic fight-or-flight state, your brain shuts down higher-order learning.
It's just trying to survive.
It is focused purely on immediate-based survival. It is not interested in forming nuanced new memories. It is not interested in neurogenesis. And it is certainly not going to engage in deep emotional therapy. It just wants to escape the perceived tiger.
Right.
By using structured breathwork to force the nervous system into a parasympathetic, profoundly calm state, you are unlocking the biological gate. You are manually creating the necessary permissive environment for the TRKB receptors to open up and for molecules like BDNF to actually get to work doing their vital repair and remodeling on your existing circuitry.
So bringing all of these massive concepts together, closing the loop on our entire exploration of mental health, the big picture of this deep dive is that fixing a broken brain or optimizing a healthy one requires an incredibly holistic, multi-layered approach. You cannot simply fix a complex psychiatric disorder by blindly dumping random naive stem cells into an adult brain and hoping they somehow find their way.
No, like pouring wet cement into a crowded skyscraper.
Exactly. You cure the disease by deeply understanding the highly specific, incredibly intricate microscopic circuitry that is failing, which is exactly the pioneering, miraculous work Dr. Paca's lab is doing with these human assembloids.
They are finding the exact broken calcium channels and designing perfectly targeted drugs to fix the hardware.
But on a personal, daily, practical level, you maintain and upgrade those exact same circuits by managing your metabolic health through strategic fasting, aerobic exercise, and diet to aggressively trigger the release of BDNFs.
The fertilizer.
And most immediately, most accessibly, you use the rhythm of your breath to control the state of your autonomic nervous system. You manage the CO2 bouncer at the door, ensuring your blood vessels are wide open and your brain is in the optimal parasympathetic state to actually receive and utilize that miracle grow.
It is the ultimate synthesis of microscopic cellular biology and conscious human behavior. The laboratory models show us exactly how the intricate machine of the brain is built, but our daily rhythmic habits determine how flawlessly that machine is allowed to run.
Which brings us to the end of our incredible journey today. I want to sincerely thank you for joining us on this expansive deep dive. We went from the microscopic sci-fi reality of customized human brain circuits literally reaching out and wiring together in a Stanford laboratory dish, all the way to the daily, rhythmic, invisible power of your own lungs expanding and contracting in your chest right now.
It serves as a profound reminder that we are highly active participants in our own neurology. We aren't just passive passengers locked in a skull, we possess the manual steering wheel.
And I want to leave you with a final provocative thought to mull over, something that perfectly bridges the two halves of our deep dive today. We've talked extensively about how Dr. Paca's lab is currently growing patient-specific neural circuits in a dish to test which psychiatric drugs work best to stop seizures or correct autism hardware. But what if we push that paradigm even further? How long will it be before scientists use these personalized assembloids to test non-pharmaceutical lifestyle interventions?
That is a staggering, fascinating frontier for human performance.
Right. Imagine a future where a doctor or a performance coach doesn't just prescribe a generic pill. Imagine a world where they take a tiny swab of your skin, grow a living replica of your exact unique cortical circuitry in a lab, and then subject your specific brain tissue to different metabolic states.
Wow.
They test exactly how your neurons respond to the chemical signatures of a 16-hour fasting window versus a 12-hour window. They measure the exact BDNF surge generated by a specific high-intensity exercise protocol. They monitor the specific blood chemistry created by 20 minutes of 6-8 breathwork.
Total personalization.
They could perfectly, scientifically map out the ultimate, flawless, hyper-personalized regimen for your specific cognitive enhancement, proven on your own living cells, completely outside of your body.
The absolute precision medicine of lifestyle and mental health. It is entirely within the realm of theoretical possibility based on exactly where the stem cell science is heading right now.
But till that science fiction future arrives, you don't need a Stanford laboratory to start rewiring your circuits. The X-ray of the mind might still be muddy, but the tools to clear the water are already built deeply into your biology. The power to optimize your mind, to forcefully calm your circuitry, and to trigger your own neuroplasticity. Wait, no, the power is literally right under your nose.